Hello friends and welcome to my YouTube channel Veteran Pharmacist. Today I am going to cover one topic of biotechnology, that is genetic recombinations. Most of the students are facing problem in this topic, so I'll explain it. The genetic recombinations, for understanding the genetic recombinations, firstly you have to understand about the linkage. Okay, so for the linkage, that is physical associations of gene over one chromosome, it is known as linkage. Okay, when from one linkage your gene is attached, okay, or you can say from one chromosome your gene is attached, so that is known as linkage, because there is a link between your gene and your chromosome, so that is known as linkage. If we are specifically talking about the recombination, so recombination is exchange of gene from one chromosome to the another chromosome. Okay, so it is known as recombinations. So you are having the one chromosomes and you have your genes over it, and the another chromosome which is also having the gene over it. When they transfer from one chromosome's genetic material to the another chromosomes, that is known as recombinations. You are recombining two different chromosomes with the help of genetic material. Okay, so that is known as recombinations. It is sometimes also known as crossing over. The thumb rule which you have to keep in your mind during the recombinations is, if the recombinations is more, the linkage will be less. Okay, if something is bind with the strong bound, then it is not able to be broken down or to be transferred from one side to another side. So the thumb rule is that if the linkage is less, then the recombination is high, or you can say that if the recombination is less, your linkage will be high. Okay, now the gene expressions. So the gene expression is the productions of encoded protein from synthetic RNA. It is known as genetic expressions. You are expressing something. Okay, so how the gene is expressing their self, that is known as gene expression. If we are talking about the gene expression, so gene expressions is mainly divided into two parts, that is DNA transcription and DNA translations. If we specifically talking about the DNA transcriptions, so it is occur in eukaryotic cells and it is occur in nucleus. The site of DNA transcription is nucleus, whereas the site of DNA translation is cytoplasm. So DNA is used as a template to make mRNA. Means the transcription is the process where the mRNA is produced. The template to make mRNA by the help of dRNA, with the process which is followed, that is known as DNA transcriptions. And whereas DNA translation is where the polypeptide or the protein synthesis mainly occur with the help of mRNA, or you can say the messenger RNA, that is known as your DNA translation. Now the next one is, if we are talking about the transcription. Okay, so transcription is a DNA is used as a template to make mRNA. Okay, as I have already told you that in the transcription there is a formation of mRNA. So the DNA is helpful in the formation of mRNA with the help of DNA polymerase. Okay, so DNA polymerase is the enzyme which is helpful in the synthesis of your mRNA. That is known as your transcription process. Okay, so transcription process, if we go into the detail, the transcription process is divided into initiations, elongations as well as terminations. So first one is your initiations. Initiations is the promoter region of gene act as recognition site for RNA polymerase. Okay, the RNA polymerase. Okay, I have told you that the polymerase enzyme is basically helpful for synthesis of the different polymerase enzyme is helpful for the synthesis of the mRNA. Okay, so polymerase enzyme. So RNA polymerase is basically combined with the gene. Okay, so gene is having the promoter region. Promoter regions, or you can say your identification region, through which the RNA polymerase is actually identified that this is the gene and I have to bind with it. Okay, so or you can say that this is the binding site of your gene. So on this they bind together, RNA polymerase on the promoter region of gene. Okay, due to that binding of promoter region of gene to RNA polymerase, double helix of DNA become unwind. Okay, when they both bind together, when the promoter region and gene, promoter region of gene and RNA polymerase bind together, they have to, DNA have to become unwind. As have you already understand about the DNA structure, that DNA is having the double helix structure. They bind together, okay, in the winded form. So they become unwinded, means they have divided into two parts. Then the elongation is the process the RNA polymerase slides along the template DNA strand as a complementary bases pair up. Okay, or you can understand that RNA polymerase is binded with the help with the nucleotides. The different nucleotides are adenine, guanine, cytosine, thymine, uracil. So the RNA nucleotide with the DNA nucleotides combine together in the process known as elongations. Okay, the third one process is your termination process. So termination process is started when your reactions is reached on terminal region. Okay, there are two basic reason, that is primary region and terminal region. Primary region is the binding site, the initial binding site where your RNA polymerase bind together with gene. Okay, so that is your primary site or promoter region. The terminal region is the ending of the process. Okay, so that is known as your terminal region. So terminal region is when RNA reach to the terminal region of the gene. Okay, this is your primary region and this is your terminal region. When the RNA reaches from the primary region to the terminal region, that process is known as termination. This means you have to terminate something. So for terminating, they have to broken their linkage. Okay, the linkage is bound up into the first step of initiations, and in the last one they are separated with each other. So gene and mRNA are separated or dissociated with each other. The various exons and introns. Exons are coded materials, introns are non-coded materials, but both are helpful in the formation of peptides. Okay, so that's why it is known as codon or non-codon. So if we are talking, that all was about the transcription. If I'm explain about the translation, so translation of message from mRNA into protein, well, it is happened by codon. I've already told you that codon is basically the non-codon and codon. Okay, and tron and exon. Okay, that is known as codon. So that is nitrogenous bases are grouped into the three letters from known as codon: A, G, U. Okay, adenine, guanine, and uracil. All nucleotides are bind together. The three nucleotides are basically bind together to form one codon, and there are different codons are used in this process. Okay, so the first codon, the example of one codon is given here. So the one codon is useful for the starting the process. So that's why I have written here AUG, that is your adenine, uracil, and guanine. The first codon which is going to start your project or your process, and the last one is UAG, UGA, UAA, or you can say any kind of nucleotide codon. Okay, as I've already told you that three different nucleotides are used to prepare one codon. So three codons are basically used for stop your codon. So one codon is essential for starting the process, whereas the three codons is essential for ending the process. Now the translation step by step, how the translation is occur. Okay, so ribosomal subunits bind together to start the codon. Okay, the ribosomal units are bind with the codon, and complementary base pairing is occur between the codon of mRNA and the anticodon of the tRNA. After that, RNA molecules bind to start codon, known as translation complex or initiation complex. In the elongation step, the RNA molecules show base pairing of the codon and anticodon. Each anticodon have specific amino acids that particular amino acids bind with A side, and the peptide bond is formed in P sides. Okay, if in the simpler language I will explain you, so I will explain through the diagram. Okay, there are three sites, that is E, P and A sites. Okay, so that all are three ribosomal site, and this is the structure of gene. It included 5 prime site, 5 prime is for primary site, where the bounding is actually occur, that is primary site. And 3 tail side means telling side, the ending one, that's why it is known as the 3 tail sites. So EPA all are the ribosomal sites. Okay, so I was telling you about the binding of codon and anticodon. So the binding of M codon or the messenger RNA codon along with the tRNA anticodon is done in this process. Okay, so that is occur into the P site. Okay, the codon of mRNA and anticodon of tRNA bind together. Okay, when they bind together, the process of translation is started. But here are two sides, that is A and P also. A and P are free now. But when the new tRNA bind to the A site, the anticodon of tRNA just moved from here to here, from P site to A site, okay, for binding with the new one. So what is actually happening? The transfer of your codon from P site to A site, and the remaining anticodon is transferred to the E site. So P is remaining now. Okay, so this process is repeated again and again and again. Whenever the new tRNA is come, it is just binding with the A site, and the transfer of anticodon of tRNA is done from P site to A site for binding the tRNA anticodon with the mRNA of A site. And the next process is done again and again when the new tRNA come, and this process is only ended at the step where there is the end stop codon is occur, means where there is a end of site is occur. When this side is occur, this is the translation process is basically completed. So hope students you all have understood about the transcription and translation process. Thank you.